While you're working on that, I'll go ahead and introduce Preetha. Preetha and I are lab mates in the Drucker Institute, and she'll be presenting um, biomarkers of disease activity in juvenile dermatomyositis, focus on B cells and T cells that lack follicular markers. So once that is all good.
Thank you, Thomas. I wanted to first express my gratitude to the organizers for giving me the opportunity to present my work, present our lab's work here in MetroFlow. And I wanted to congratulate Peter. And I had had the opportunity to work, not work, collaborate with Peter and during my graduate project, but he might not remember that. I was in Mike Dustin's lab before, and it's really a great honor to talk after Peter. And so today what I'm going to be presenting here is a work that I collaborated with a physician scientist in our lab. This is a very collaborative work. And as Thomas mentioned, I'm going to be talking to you about biomarkers of disease activity in juvenile the dermatomyositis, focus on B and T cells that lack follicular markers. I work in Virginia Pascoal's lab. So this
There we go.
Sorry about that. Thank you, Thomas. So this paper was published earlier this year, and I worked closely with 3 physician scientists who are lead authors in the paper to develop the panel for this paper. So to give a brief introduction about JDM, it's idiopathic inflammatory myopathy that you see in a pediatric population. It is characterized by muscle weakness and skin manifestations and systemic vasculopathy. You can see in this image how kids show up in the clinic, and they usually show flares and skin rashes, and you also have this characteristic rashes in the fingers as well. So the disease is very heterogeneous in clinical manifestation and its response to treatment, as well as in the clinical prognosis. So what are the treatment that are available right now is mainly immunosuppressive drugs. And you can see in this graph how the disease is heterogeneous on the top graph. And the main way they are treated is very much a trial and error approach. And so there is a need for biomarkers, because once we identify biomarkers, then we can stratify patients based on the heterogeneity and have a targeted approach to treatment. Now, coming to the disease pathogenesis, what we know about disease pathogenesis, the first thing is autoantibodies. In JDM patients, we do detect autoantibodies, and there are 2 main classes of autoantibodies that are detected in these patients. One is the myostatus-specific antibodies, and another one is myostatus-associated antibodies. There are 2 different kinds, and there is a graph that I've shown here, which shows the different percentages that are present in different patients. And as I mentioned, it's heterogeneous. And so there is another feature that is very distinct in JDM patients is the interferon signature that is detected. And this is present in blood, skin, and muscle tissues. And also the interferon signature that is present in the blood is associated with autoantibody that is found in the serum. Now, as I mentioned, autoantibodies, everyone is very well aware that T and B cells play an important role in antibody generation. And what you see here is a graphical representation. On top is the classical T follicular helper cells that help B cells. We know in settings like SLE, systemic lupus erythematosus, that there are circulating T follicular helper cells in the blood. that are expanded in SLE patients. And of course, that is a classical pathway, and we think that that is one of the causes for autoantibody generation. But recently, there are several publications, both in rheumatoid arthritis and SLE, that show that there are other peripheral T helper cells that help B cells, and that too B cells that are double negative B cells, that double negative B cells that are not the conventional memory cells or the conventional germinal center cells that are found to be expanded in SLE. And these T helper cells are found to be expanded in the synovium of arthritis patients, and they have shown in these studies that these can differentiate, these cells can help in the differentiation of these double negative cells into plasma cells that produce autoantibodies. Now, a study that I contributed from the lab, we characterized another T helper cells in the SLE patients, where we show that cells, unlike the classical IL-21 and CXCL13, we show that the IL-10 and succinate play an important role in inducing the double negative 2 cells to generate autoantibodies. Now, coming to JDM, what do we know about T and B cells in JDM? There are papers and reports that have shown that total B cells have expanded in JDM patients, and there is also expansion of what we call as the transitional B cells. And here in this setting, they've also found that TLR7 induction, TLR7 signaling in the transitional B cell led to a more pro-inflammatory phenotype. And there is another study that showed that there are T follicular helper 2 and 17 cells. So the conventional TH17 and TH2 cells that we see in the periphery. Similarly, there are classes in T follicular helper cells which are CXCR5 positive, and they, of course, secrete TFH2 secretes IL-4 and help in more IgG, IgG antibody generation, whereas TFH17 cells more of the IgA. And what is shown here is that they found in patients in the JDM patients that this ratio of TFH2 and TFH17 is much more than the TFH1 cells. Now, what we tried to do in this study was to ask if we can characterize the T and B cells by flow cytometry, and also look at transcriptional data from these patients and see if we can correlate the cellular phenotype with the transcriptional data. This is the overview of the study. We had an initial cohort of JDM patients, and of course, here you have healthy controls in both the cohorts, and the main way we distinguished the patients was based on the patients were either inactive or active JDM patients, and this was based on the clinical assessment of the physician for muscle activity. And this was subjective, of course, but any score of above 0 for more than 2 years were classified as inactive patients. And so the initial panel for the initial cohort was only 11 color panel. It was mainly focused on T and B cells, but then the validation panel had a bigger 25 color panel, and we had all the clinical disease scores for all the patients, and of course, this followed by analysis. For the transcriptional data, we had another cohort where we had an additional control of 4 SLE patients, and we performed bulk RNA sequencing, and we also had the clinical information to stratify the patients. Of course, I don't have to say for this audience, a lot of people do flow here, but this is, we had an initial panel which had only the T and B cells that was focused on T and B cells, and then later we expanded the panel to a 26 color panel. It was also because initially we had a 3-color laser aurora, and then later we had a 5 laser aurora that we bought, and so we were able to expand it. Although in this paper, the main focus was only T and B cells. So I will be only showing the immunophenotyping of T and B cells. So here in the panel, I have just shown here what were the markers that we used for characterizing the T cells. And this is just like a flow of how we gated on the CD4 cells. And here I would like to spend a few minutes, a couple of minutes just to explain the T follicular and the TH, the conventional TH, how we gated the cells. CXCR5 positive cells, which are CD45RA negative, where the blood TFH cells that are then classified based on the CXCR3 or CCR6 expression. The ones which are CCR6 are the TFH17, like how we generally do for the TH17 cells. And TFH2 are the double negative cells here for CXCR3 and CCR6. Now, well, when we did look at the analysis for both the cohorts, what we observed was that we were able to recapitulate what was earlier found for the ratio of TFH2 and TFH17 to TFH1 cells. And this was specifically more expanded in the active JDM patients compared to the healthy controls. The frequency of the TH1, the conventional TH1 cells, which are CXCR3 positive, was contracted or decreased significantly in the JDM patients, again in the active JDM patients. Although the TH17 cells were not significant, we didn't see any expansion of TH17 cells. And what was really interesting was that we found an expansion of TH2 cells. TH2 cells that are CXCR, the conventional CXCR5 negative TH2 cells. And we also found that within the active JDM patients who are diagnosed within a year of, 1 year of diagnosis, there was more TH2 cells compared to those which are more than a year. And then moving on to peripheral B cells, the panel that you have shown here is to characterize the B cells. And so the gating here is on CD20 positive cells. Of course, live single cells were gated, and then CD20 positive cells. And then we use CD27 and CD38 gating to remove the plasma cells. And then we further gated using CD27 and IgD. And here the conventional memory cells were further gated on CXCR5 and CD11C. So this gating was to look to find if there is CXCR5 expression, to see if we find like extrafollicular B cells. And what I wanted to point out was in SLE patients, we find an expansion of the double negative cells, which are CXCR5 negative. So initially we were hoping to see if there was an expansion of double negative cells, which are CXCR5 negative here. And you can see this is a healthy control, and this is a representative gating for the JDM patients. And you can see that there is a population of cells here which are CXCR5 low conventional memory B cells that are much more compared to the healthy controls. So the first thing is, we found CD19 cells to be significantly more in the JDM patients. And the transitional B cells, like that was seen before, we found that transitional B cells were expanded in active JDM patients. There was an expansion of naive B cells, but there was a contraction of the conventional memory B cells, which are CD27 positive IgD negative cells. The interesting thing, as I pointed out, was the cells that are CXCR5 negative, the conventional memory cells which were contracted, but then those that are negatives of CXCR5 were much more in the JDM patients. And we saw that this was true in both the cohorts, and as well as it was significantly more in the active JDM patients. And this is the absolute count of these cells were also significantly more in the active patients. And it was very interesting and striking that both the TH2 cells and the CXCR5 low cells that I just showed were both associated with the aldolase. So the lab aldolase, this is a muscle activity enzyme that is detected in the patients, and both were correlated or associated with the expansion of both TH2 and the CXCR4 conventional memory cells. Moreover, we also found that the muscle activity, which is scored by which is shown by MMT8 and CMAS, were both associated with the absolute count of the CXCR5 low conventional memory cells. And so and finally, we found that the CXCR5 low conventional memory B cells were associated with the expansion that we see for the Th2 cells. Now, this brings us to what might be the interaction between these cells and the Th2 cells, which we are interested in characterizing right now. This is something that is ongoing in the lab. Now, moving on to the transcriptional data, as I showed, there is a third cohort that we had. We looked at the bulk RNA-seq, and what I would like to bring your attention to is this distinct interferon signature that we know that is higher in JDM patients. And JDM is an interferonopathy, and we know that type 1 interferons are much higher in JDM patients. And you can clearly see in orange are the active JDM patients, and you can see that it's much upregulated in the active JDM patients compared to the inactive patients. And there are other inflammation-related pathways that are again upregulated in the active JDM patients here. And there are erythrocyte-related modules here, which are again distinctly more in the active JDM patients. When we stratified the patients based on this particular module, this module 1, M1.2, there's an interferon module for type 1 interferons, we found that the active JDM patients were much more higher. They showed a higher module score compared to the inactive patients. And finally, when we looked at the correlation between what we found expanded in the T and B cell compartment to the interferon scores that we found that the type 1 interferon score were correlating with the expansion of both the CXCR5 low central memory cells and the Th2 cells. To conclude, to summarize what we found in this paper, we think we were able to consistently see that the CXCR5 low conventional memory B cells correlated with a high muscle disease activity, and it could serve as a disease marker. And the expansion of these conventional memory B cells that are lacking the follicular marker correlated with the Th2 cells in the periphery. It also showed a correlation with the interferon signature. Now, type 1 interferon signature it is known, it is understood in the literature, it is shown in the literature that type 1 interferon can induce more B cell signaling, and it was also shown that transitional B cells due to this interferon signaling was showing more pro-inflammatory phenotype. So it would be interesting to characterize these cells to see what is going on with respect to the interferon signature that is correlated here. This is a snapshot of what we think is going on. Our lab also works with lupus erythematosus, and we know that the ratio of Th1 cells, the Th1 cells are more expanded in SLE patients, and the DN2 cells, which are the extrafollicular B cells that I showed before, is expanded. And we know that it is related to autoantibody generation. Now, coming to JDM patients, what I just showed was that the Th1 cells are low, whereas the Th1 to Th17 ratio again is low, whereas the Th2 cells, in another study, we characterize the Th2 cells and we show that these are expanded. And with respect to B cells, although we do not see an expansion of DN2 cells, we found that this CXCR5 low central memory cells, conventional memory cells are expanded. And would this be related to autoantibody generation is something that we are interested in understanding more. So finally, this is something that is ongoing, but we are really interested in looking at single cell, at the single cell level, the B cell compartment in JDM patients. And we have already performed single cell sequencing on a cohort of JDM patients. And we have some preliminary data that suggests that we can distinguish the memory compartment we have a very well-established analysis pipeline where we are able to subcluster the memory B cells, and we identify 3 different clusters. And so we think that whatever we see at the flow level, we kind of recapitulate that at the single cell level. And we also try to add on based on our single cell profiling if there are additional markers. So for this work, it was mainly led by the 3 physician scientists, and Thomas really played a huge role in when we expanded the panel. And he's always been there to, he's very encouraging. He's there in the lab to help us all the time. And I also work closely with Marina, who's here, and Owen in the lab also works with us. He's here right now. I would like to thank my advisor, Virginia Pascal, for giving me the opportunity, and work closely with Simone Kayali in the lab, whom I have done a lot of work. I have collaborated for a lot of work. And thank you for listening, and would like to take questions.
Great job, Preetha. Any questions?
Thanks. Great presentation. This is Amritesh. So you found the origin of type 1 interferon in JDM as against lupus?
So it is already known that there is a high interferon signature.
No, you found the origin of type 1 interferon is
No,
are
we
the
don't know the
Th2
source.
cells?
You mean the source of the cells?
Yeah,
The source of cells that, no. So that was a bulk RNA-seq, so no, it was the total whole blood.
And the interferon module are interferon stimulated genes, or it's the interferon?
So when I say interferon module, it is the interferon stimulated genes. Yes.
So you didn't find IFNA or IFNB, it's the interferon
No,
stimulated
it is
genes, which sort of is a surrogate marker for interferon, correct?
Yes, no, it was the interferon stimulated genes.
Okay, thank you. Great presentation.
Thank you.
So I have a question, Luis Mendez from Merck. Wonderful presentation. Just out of curiosity, you went through lengths to characterize your B cells and your CD4 positive T cells, but why stop there? Why not go into and look at regulatory T cells that obviously have a central role in suppressing the immune function in both SLE and JDM?
Yeah, great question. Actually, I'm really interested in Tregs, and currently I'm characterizing them in SLE, and that's my main project. So for this particular study, our focus was actually to see if there was an expansion of DN2 cells, because that was something that we started the project with. And then, of course, at that time, we are also published the paper on Th10 cells that were expanded. So that was really like the main question. We still have the data. We are still looking at it. But at this, so we had, at this point of time, we have like characterized only the T and B cell compartment that I was showing you.
Looking forward to seeing that. And then also, obviously, PDCs are implicated in SLE for type 1 interferon. Do you suspect the same thing in JDM?
Yeah, definitely. So our single cell data kind of kind of shows some signs that it could be the case, but we have to verify that.
Any more questions? Great presentation.